All right. Mike Mahan is a professor in the Department of Molecular, Cellular, and Developmental Biology at the University of California, Santa Barbara. Mike, welcome to CC Life Science.
Thanks, Chris, and thanks for having us.
Yeah, my pleasure. So, let everybody know we're going back to my roots today. Mike and I were lab mates in graduate school in Utah, and then I worked in Mike's lab in Santa Barbara way back in the 90s, the late 90s. And today we're going to talk about antibiotic resistance following my recent episode with Randy Rohde. And this time we're going to focus a little bit more on new antibiotics, challenge of finding those, and susceptibility testing, which sometimes gives answers that might not reflect actual in vivo effectiveness. So let's start off just talk about the problems of antibiotic resistance generally, and then beyond that, the challenges of finding, creating, and commercializing new antibiotics.
Yes. Well, these are all very, very timely topics, and thanks for the opportunity to be able to discuss them. Basically, the World Health Organization has declared that antibiotic resistance is one of the major threats facing global health. Okay, some bacteria today are resistant to all or nearly all medications known. There's drug-resistant staph, drug-resistant strep, drug-resistant TB. You get these bacteria, there's nothing that can be done with antibiotics, and we need to act quickly. I mean, it's not going to happen. Well, we're there, okay, and we're entering the pre-antibiotic era, where a simple cut in the finger can lead to death, sepsis, and death, and so despite this urgent need for antibiotics, there's been very little done in the last 20 years. Okay. And the reason for it is that the pharmaceutical companies have virtually abandoned antibiotic research. And it's very simple. It's very risky and it's not profitable. Two good reasons not to do it if you're a pharmaceutical company. And so what they've been focusing on are blockbuster drugs that have a long-term time course, i.e., cardiovascular disease, diabetes, cholesterol, high cholesterol, immune disorders, cancer. That's where they've been focusing, and it's all math. It makes simple sense. I mean, it takes 10 to 15 years for a new drug to come to market. It takes 10 to 15 years of research at a cost of $1.5 billion. Okay, and what the average drug revenue is $50 million a year. That's 30 years just to get what you put in. And they've I can tell you that 95% of all antibiotic research is not done by pharmaceutical companies. It's done by biotech companies. And a lot of them have never produced a product. The pharmaceutical companies, they have the means, they have the mode and the opportunity to get it done. And so there's so we're trying to figure out, we have to now basically put a different value on antibiotics. Okay, that they're worth a lot more than what the revenue says, and so as a society, you're going to have to come to that conclusion, in my opinion. And there's these new ideas that have been floating around for a while. Is that have the government provide pharmaceutical companies for R&D funding. Say, we think this is important. Here's a sum of money so you do work on antibiotics. And the second is to give what are called subscription fees. And so we're just going to have to change the way we view it. I mean, it can't be all on the pharmaceutical companies. We have to, as a society, have to say, okay, we're going to have to pay more for these. I think antibiotics are important. And if one of my friends or family gets infected, I want them to get an antibiotic that'll work. And so hopefully this will start a conversation. It'll fuel the conversation to be able to actually do something meaningful. And so, how do we get here? Basically. Well, in 1966, the World Health Organization made a decree or declared that we're going to test antibiotics a certain way. We're going to use this one media. The whole world will use one media. And it made sense to be able to test antibiotics because apples to apples, like if you're infected, in the Bay Area versus Santa Barbara versus Africa, South America, et cetera, we all are using the same test. It made a lot of sense. And they chose a specific media. It doesn't matter the name of it, but it's a rich media, like chicken broth, basically. And it was chosen because it was super cheap, it's widely available, and many pathogens grew on it. 3 really good reasons. There has not been one change since 1966. It's almost 60 years. And Chris, you and I are geneticists. That we've been thinking about this for a while, and this makes no sense from a genetic standpoint. That why would a rich media be the standard? It always something in my mind was always going on. This has been for a really long time for me, and if you can now mimic the body, the bacteria, the action of the antibiotic against the bacteria in conditions that reflect the body may more accurately predict the patient outcome. Okay, and this has been something that's and why hasn't it been changed? It's really hard to change. What I've learned that the difference between science at the bench and actually in the clinic, it is palatial. It is not. I made this discovery. No, that's not the way it works. And actually, after we made one of these discoveries, that discovery I was going to tell you about a new antibiotic susceptibility test, I got in touch with with UCLA, which runs this very large facility for any new antibiotic test. And so, yeah, I read your paper. That's great, And he said, but you know how hard it is to have the world change their test? And I just kept saying, but we both know it's not good enough. And I just kept saying that for an hour. He finally just gave up. He just caved and said, okay, when you got it, get a hold of me. And I haven't done it yet, but that was in 2017 actually.
Wow.
But anyway, so let me tell you about this new antimicrobial susceptibility test that we've come up with. And basically, the solution was to be able to define a media that more reflected the body. And bacteria, when they're in the body, they sense and respond. They'll change their surface, they'll change their gene expression, their protein expression in response to the environment they're in. And we thought that may more accurately reflect when you're challenged with antibiotics. They sense and respond to this, and they respond very differently. And what we showed was that using this test, that we've now checked FDA-approved antibiotics that are dismissed by the gold standard test. And the answer was there were several antibiotics that were highly effective in treating sepsis in mice that are never used by physicians because the gold standard test says it won't work. And so consequently, our toolbox for antimicrobial infection is much larger than we once thought. Yes. Yeah. So anyway, the bottom line is that it's very simple. People want Petri plates. Okay, that's why antibiotics fail. Okay, and this research has 2 major implications. One is that it optimizes the prescription and use of current antibiotics. Number 2, it enhances the efforts to discover new ones. So a new test can be used to bring in the antibiotics we already have and also discover new ones. And if you can get, and the whole issue is, can you accurately predict patient outcome quicker? These trials are expensive. Okay, and what we showed actually that 2/3 of the time both tests, the gold standard test and our test agree. 2/3 of the time, great. And about 20% of the time, there's a big difference in the fold of how sensitive or resistant they are. But 10% of the time, it crossed what's called a clinical threshold, and that's what physicians, that's what they look at. A clinical threshold means resistant or susceptible. And so what I'm saying is 10% of the time, the drugs we tested, and we tested a lot of drugs and against a lot of pathogens, that the gold standard test said it would be resistant, our test said it was sensitive, or the gold standard test said it should be susceptible, and our test said it should be resistant. And we tested it in an animal, and in a very large animal experiment, the answer was the new test accurately predicted markedly better than the gold standard test. But on the positive side, when you did both tests together, it was nearly 100% correct when they agreed. So when they both agree, it's like that would give some confidence to physicians. That's all we're saying. And this test is super cheap. There's nothing different than the standard test that's been used globally. It's just a different media that's also super cheap.
Yeah.
It's cell culture medium. Okay. And I can tell you the physicians have known that this test has been flawed for a long time. Okay. And what they've done, here's an example. The Z-Pak, which is this zithromycin, it's the second most commonly prescribed drug in the United States. You give it to your kids for ear infections, and it's very powerful. Okay. However, against sepsis, bacteria that cause sepsis like MRSA, E. coli, strep pneumonia, things like that, it comes in last, basically on the gold standard test. Don't use it for that indication. Use it for skin infections, ear infections, whatever. However, in our test, it's at the top. Okay, it says yes, it will work. And anyway, physicians have known just by their experience, they've been going against the test and prescribing azithromycin for sepsis, for MRSA, for instance, other nasty pathogens as well. And this is the minority of them, but there's some ones that have been very vocal about it. No, I'm sticking with this because it works for me. And however, if you're a physician, at every major medical center in the country, there's an antibiotics use committee trying for antibiotic stewardship. I.e., you can't just give everybody a re-antibiotic because we're not going to have any left. Okay, so it makes a lot of sense to have these, but you can't just go. We call it go rogue or go off menu. You have to actually. You're a physician. You can do it, but you have to justify. You have to now say, "I'm using this because this is my experience," and you got to do that almost every time that you do it. And here we're given some, hope for the disconnect that physicians know is there, but they're conflicted. Okay, should I do what the standard says to do, or should I do this other thing that's based on my experience? That's a hard call. Okay, and this gives so every physician actually since this paper's come out it's been all positive. Like, Mike, this is great. I mean, we cite your paper, and it's so I don't know.
That's great. I want to ask. So I have a bunch of questions just around that. The new media, the non-gold standard test.
Mm-hmm.
How did you think about what that should be, or did you just imagine, all right, here are my selection of growth media. I think this one is a better choice than rich media.
Yeah.
I'm just curious about that.
It was simple that the simplest thing we could think of to reflect the body was to use what's called cell culture medium, which was designed to grow mammalian cells. The rich media is designed to grow bacteria. Well, that isn't, it was just a disconnect forever, so it was the first thing we tried and it worked. Now we're actually trying, in the paper, we've all tried, human sera, human urine. As of today, the best media has been this cell culture medium, which was surprising. Yeah. But we have some reasons why we think we can tweak those as well that we're working on.
Yeah. But, so the light bulb just went off for me. I mean, of course, right? I mean, in retrospect, of course, not like de novo. I wouldn't have thought of that. But
Chris, you wouldn't. But you were my minister. But you're the smartest guy in the lab. So,
on the other, thank you. But on the other side of that, cell culture medium. So, I guess if you have that, so now going the other direction, why would you change it if the in vivo experiments on the mice correlate very well with that cell culture medium, the new test? What are you looking for in a different test, except maybe to tease out gold standard things that are on the edge and which way should we go or something like that?
Great question, actually. So even though it was markedly significantly better than the gold standard test, and I mean, I had to say this, it had to be, or there'd never be a change, right? Our first paper on this was in 2015. Nothing happened. 2017, we only did one, salmonella and a couple drugs. We thought, this is going to change the world, nothing happened. And then in 2017, we did, 20 pathogens, 20 drugs, but not that many mice, different animal models. They're hard to do. And this time that came out, this year, a couple months ago, that we did, a very large animal testing experiment, which is really hard to do because these animal models are tough. And as Dr. Heitoff, that runs my lab, that I know, Chris, that you work together, he's really good. And he has the capacity to test almost any animal model. And so we tested very large animal models and showed that statistically, and we set the statistics against us. We used the most advanced. This isn't biostatistics. It was statistics for statisticians, which is a different thing. We weren't pea hunting. Let's put it that way. We were okay. We set ourselves up, to the most rigorous standards we could, and the answer was it was highly significant. Okay, so now to your answer your question is that, but it's not perfect. Okay, and I think we can do even better. Okay, and I think it's important to always evolve to perfection. I mean, if we can do better, because that's lives we're saving, right? I mean, I can tell you this, that if you're going to sepsis, every hour that you're on the wrong antibiotic or not on an antibiotic, there's a statistically significant increase in the chances of death.
Yeah.
So because what you're doing, you're having inflammation and coagulation throughout your body. A lot of that's irreparable. You have blood clots in your heart, your brain, your blood vessels, and other organs and tissues. And so you want to be able to be on the right antibiotic as quickly as possible to be able, or you can have these, life-altering events. Sepsis is a 30% mortality right now, the best medical centers in the world, 25 to 30. However, in survivors, many of the survivors have long-term cognitive decline, i.e. due to this clotting and this inflammation that occurred. That's not repairable.
So, I mean, it's good to hear, all those stories, and the physicians are starting to get it. Before we shift gears to talk about a new antibiotic, what do you think it will take for widespread adoption of something other than the gold standard?
I think it's going to be really hard, actually. And just with this experience from this person at UCLA, that he's right in the middle of it. And he said, to change this, do you know how hard this is going to be? And the only answer I had is, well, we can do better,
Yeah.
And however, we're running antibiotics now. When we first came out with this in 2015, life's different in 2023. A lot has happened. And, really, we're running out of drugs. And it's every day now. It's not like, oh, yeah, somewhere in some place, no, it's at your local medical center to which you go. They're seeing this, and now it's the timing is much better than it was. And the thing is, I get it. In my view, I said, our medical system, let's speak for the United States, we're great in catastrophes. That's where we excel. COVID, we excel in that. That's our strike zone. Okay, but to prepare and stuff, that's not, because it costs money and it costs time. And so, but to be fair, let's be fair that you're doing all these, but you're doing something else other than the antibiotics. Do you know what I mean? Like, they're doing stuff, but it's not this. So it's not a gag. It's just saying that you have to get on the top of the table. And something like your show here, this actually helps us to get the word out that this is not something in the future. This is happening in every major medical center in the country.
Right. So. Yeah. It's similar to what Rodney Rohde and I talked about, the victims of our own success. Most of us, we've grown up in the golden era of antibiotic or lack of, I won't say lack of infections, but my parents' generation, your parents' generation saw kids die of infections. Most of us have not had that experience. So it's not, we just take it for granted that, oh, you go to the doctor, they give you a pill and you're done. But as you point out, we're heading back to the we're going to be looking at that. And what a tragedy in a way to think like, all right, for 80 years was the window in which we were able to do this, but we're going to find out what it was like in 1910 pretty soon.
Yes. It's a scary thought, and I don't want to be Johnny Raincloud here, but it's the reality that we're entering. And I think that for some reason it's not out there to the public that the alarm bells have been ringing for a long time. They're wrong now. It's here, and life, if we don't change things quickly, act really quickly, which I think we will. So I think the answer is we will. That we're gonna dive back into the pre-antibiotic era, and then you just don't make a new antibiotic. Those things are hard. As I've said, we've really had one major class of antibiotics since 2000.
Yeah,
that's not exactly hitting the homer here. It's like, and the reason, there's a lot of reasons for it. The abandonment of the pharmaceutical industry, also litigation. It's much easier to make another penicillin that we know is, penicillin is very non-toxic. So that's why it's a great drug. Now things are resistant to it, of course, but many of the new drugs are penicillin derivatives these fancy new carbapenems and cephalosporins. They're penicillins, okay? And the reason for it is that, a lot of it is that we know the toxicity is low. And, you don't want to be sick from taking the antibiotic, right?
Yeah.
Yeah. And so that's always one of the hard things is the toxicity. So, so for litigate, you just can't take that chance, and they're not taking that chance. That's the evidence in the last,
Right.
So anyway, but I can tell you that. So about where you asked to your question about, where are we now? What's next? Well, what we're going to do is that it's called adjunctive therapy. So basically, what they're going to allow us to do is not replace the therapy. So let's say if you're on penicillin and maybe azithromycin, we believe is the correct drug. If the patient is not doing well on the penicillin, they'll allow what's called adjunctive therapy or replacement therapy. Sorry, not replacement therapy, but additive therapy. You add this to the cocktail. So it'll be penicillin and azithromycin. And if that works, then they'll be studied. They'll allow us to be able to do, that's going to take some time. And then they're going to say, okay, now we'll do experiments on replacement therapy. Instead of doing penicillin, we're going to start with, let's say, azithromycin. Does that make sense?
Yeah, I got you. That's
so it's a road. It's a road, but that road
is a road.
That road we can do, actually. Yeah, I see the end of that, actually. So that's but a new antibiotic, the road is much harder. Okay, we're talking about stuff where these are, the FDA approval is already there. It's much simpler, even that. But we're dealing with humans now. I mean, let's just we got to be careful. So anyway, if you want to hear about the new antibiotic that we've
let me tee this up for you. So we're going to talk about a new type of antibiotic. Nature Medicine published an article this month in which you were interviewed, talking about this is the hardest word I've ever said: conjugated oligoelectrolytes. I don't know why I've struggled with that. So referred to as COEs.
Yes.
Tell me about how you got started testing them, because they weren't really developed as antibiotics, it sounds like.
Correct.
And then, give us that story.
Yeah. So basically, the U.S. Army was a pressing need for charging cell phones. When batteries go out, so when your cell phone dies, you die. Okay, that's just the way. That's what they instructed us. And so the idea came about to be able to solve that problem, where bacteria are power cells. They make energy, right? And what they do, they make the energy into making more bacteria. And so a chemist colleague of mine was funded to, instead of the way it works is that the power machinery is in the bacterial membrane on the outside, and it now shunts electrons, which is the power, to do work, gets shunted into the cell. And he simply made these COEs, these conjugate oligoelectrolytes, to, instead of the electrons shooting into the cell, these chemicals would now reverse it and make the electrons shoot out of the cell. And he could power a light with it, with bacteria. So the idea would be to have like a Gatorade backpack where, you could live in a cave or the jungle or whatever for, a Gatorade basically providing sugar to the bacteria, and then the bacteria now providing electrons to be able to power your phone or your light or whatever. And actually it's working. Okay, so that's what the original funding was for. And then the idea later, it had nothing to do with the funding, that with the reason we were starting with this, came, well, maybe you can use these for antibiotics, because we noticed that it wasn't great for the bacteria when they were.
Yeah, that was my thing. Like, how do you keep this thing alive? How do you keep the bacteria alive and working for you?
Well, first, I'll tell a kind of funny story. So when the chemist came over and asked me, he asked me nicely, and I met with Doug, who runs my lab, and we met in the conference room, and anyway, he told us about this story, whatever. And I actually literally kicked Doug under the table and going, "This is going to be a really short collaboration." I said, "This is like going to be freaking bleach, man. It's going to be so toxic that bleach is great, antimicrobial, but you can't drink it." And I thought it'd be like that. And so the chemist gave us a whole variety of them, not knowing which one would work as an antibiotic. He gave us like 17, I think, was the first patch we used. And I said, okay, we'll test 17 for efficacy, which means can you kill bacteria? But we're also going to test them for toxicity, i.e., will it kill mammalian cells too? Okay. And anyway, most of them, we were right. We were toxic as hell. They just killed our cells, bacteria, whatever. But one out of the 17, it killed every pathogen we tested. I'm talking about ones for which we derived from patients for which every antibiotic at our local hospital didn't work on. MRSA, for instance, Klebsiella pneumonia, which is actually the most feared pathogen by physicians in the United States. It's a nasty pathogen, causes sepsis. It's called carbapenem-resistant Klebsiella that killed the patient. And this drug killed everything, and it wasn't as toxic as the others, like significantly less toxic. We kind of went, whoa, I was just blown away. Like, I knew they would work against the bacteria, but I thought they would be. And so I'll tell you, I'll be honest. If it didn't, the one of the 17, we would quit. We would have been other things to do. It just happened to be in the first batch we tested. Okay. So then we said, okay. And the first thing we did was, our approach is different than the pharmaceutical industry regarding antibiotics, and we go to the animal first, not last. And the pharmaceutical company, they have this protocol that they go through. I understand it.
Yeah.
Okay, so everybody in the world does it. I get it. Good for you. It's et cetera. However, the last thing they do is checking the animal because they're expensive. It's super expensive to do.
Yeah.
It requires really, really highly trained people to do it. It's not like you just don't hire somebody off the street to do an animal experiment. They have to have years of experience. I get all that. But this is our sweet zone. This is what we do every day. So we checked if it could cure. It's called refractory bacteremia, which is a fancy way of saying the patient didn't live. Okay, i.e., refractory, and that refractory to all antibiotics that were tested. And one was a MRSA, and the other was this carbapenem-resistant Klebsiella pneumonia. And we infected the mice. We gave them 1 dose a day, and we cured them. Okay, and these are ones that, especially for the Klebsiella pneumonia, that's kind of an interesting story. It came from a UTI patient in the Santa Barbara area, had a UTI from the community, and they gave the standard drugs, didn't work, got sicker, didn't work, got to the hospital, gave them, didn't work, sent it to the other area hospitals, none of them worked, and she died. She died of the infection and dying of a UTI. Okay, this is from the community. I get it, people. The world, the scientists get it. If you're in a hospital, you're around a lot of sick people, you have a lot of the latest antibiotics, you're just there. Now from the community, that means something's, the cat's out of the bag. Okay, and actually that's how we got funded. We went to for this work. I told the story to the head of infectious diseases. It's called RARE, the Walter Reed Army Institute of Research. That's where the president goes if he or she is sick, if they can make it. And it was the first week. This guy, he's the head of infectious diseases, and somehow we got an hour or two with him. And I told the story. He didn't care about the chemicals. He didn't care about it. I just said, this is what happened. And he was from Stanford, actually. He's a professor from Stanford before he took this position. And he goes, anybody on my watch dying of a UTI of any places in the world, Santa Barbara? I go, this is unacceptable, and I want it funded. And that's how we got the money. I just went out there and shared this story with them. And anyway, the bottom line is that so now we had a drug that could kill, that was deemed, these diseases were deemed untreatable. Okay, so that's great, but the one thing that we're like key part to the finding actually as a geneticist is that the first thing we did, and Chris, we worked together for many years. The first thing you do when you have an antibiotic, you try to make a mutant. Okay, you become resistant, and then that'll tell you how the antibiotic works. Well, Doug, again, Dr. Hightower has worked in my lab for 30 years, got his PhD, been there 30 years. And anyway, starts doing serial dilution every day. First day, nothing. Like, what do you mean, nothing? I mean, I was actually not that nice about it. I just said, come on, dude. This is, I just said, this is good. I'm so sorry.
Can I interrupt here and just say, if you knew Mike Mahan and Doug Heidoff lays down cells and gives it this antibiotic and none of them show up resistant, there's something missing in the media you did, right? No way that comes out.
So he did it for 21 straight days, like serial dilution, and found nothing. That was with salmonella. And I said, okay, and I started believing him. He goes, Mike, I said, drop all your projects. We need this. We need it bad. And the answer, we couldn't get it. So we'll try another. So we tried at MRSA, nothing. So salmonella and MRSA are two different classes of bacteria, and both of them didn't. We got no mutants. So there was a place down in Sanford Burnham Research Institute, which is down in La Jolla, It's associated with UC San Diego. And anyway, this person who I didn't know, but was recommended to someone, we got an interview, with him. Actually took my son, went to Scott and went down there and said, hey, we told him the story. And what he does is he screens drugs for, I can't tell you, a major pharmaceutical company, 365, 24/7. And what they screen him for is what's called irresistibility. I don't like the word really, but that's what it's called. It means most drugs, or let's say many drugs, they work, they may even be not toxic, but the bacteria become resistant to them too quickly, so you don't make any money. Okay, so he does for a major. That's what they do. That's how he funds his lab. And anyway, so I told him the story that we have this drug that can kill every pathogen. We don't see that much toxicity. We can't find any resistance. And I said, I'm a geneticist. I've been here for 30 years. I've never seen anything like this. And you could just see it was just blank face, like, yeah, right, And
he's kicking his buddies under the table, going, "This is going to be a quick project, right?"
The problem is I needed him. We needed him. He had a fancy. Chris, you'll know what it's called. It's called a morbidistat. It's like a chemostat. Basically, what it is, it's all hooked up to computers, and you can input what media you want, and the rate in, the rate out, has all that stuff. There's only like 4 in the world. Okay, and I'm sitting in there, and I got to get in that freaking machine, So I said, you tell him just like, I know we're busy, whatever. I said, you know what? Let me just send you the drugs, send you some pathogens, and whenever that thing's open, can you, you run it for us. And I just didn't leave till he said yes. I just wouldn't go. And so anyway, so it it's going to be at least a month, 5 weeks. I said, fine, whenever you can get to it, because we didn't have another option, So anyway, so like 6 weeks later, I get this call, this text, an email, all urgent, exclamation point, 3 exclamation points, and I thought someone died, actually. I thought, because we sent them some nasty pathogens. I thought, oh, crap. It'd be a bad day, It's like, okay, all right, what's the story? He goes, Mike, this is the most irresistible drug we have ever tested. Okay, and he goes, and we've done this for 20 years. And so anyway, I said, well, I told you that 6 weeks ago. And he goes, yeah, we didn't believe you. He was honest. But then he was very... and he's a wonderful guy. His name's Dr. Andre Osterman. He's a great guy. And anyway, he's really been instrumental in pushing this along. But anyway, how can it possibly be that you can't get resistance to this drug? I mean, that's just unheard of, right? And so what we have found, and we don't know the details, but Dr. David Lowe, who Chris also worked with at the University of Utah, and now he's at Santa Barbara. This is in his strike zone. And anyway, membrane changes and things. And what we found together was that this drug doesn't affect one target. It affects many targets, many important targets that are required for the bacteria to survive. Most antibiotics, almost all antibiotics, have one target. Like penicillin affects the cell wall. Okay, you disrupt the cell wall, the bacteria, it's like a snake, blows up because it can't make the skin. Okay, well, what we found was that many essential functions in the bacteria this drug got into the membrane and affected, energy production. Makes sense because they were designed to shoot the electrons the wrong way, and also affect the membrane permeability. So crap was getting in that never, things that get in shouldn't get in. It became non-modal. It couldn't swim to a source. So all these things we believe added up to the ability to kill every pathogen we tested, but also why there's no resistance. We take multiple mutations to get there, and so we think the future is bright. That hey, we've made something that thinks going to make a difference, but it's going to be a long road. I'm going to tell you this, way longer than the antibiotic test. I see the end. Okay, that is going to happen. Okay, yeah. Change the media, and now people live. Okay, without taking any chances. Okay, and we're saying use the current test and this test. They're both cheap compared to someone dying of sepsis. That is one of the most expensive diseases in the hospital. Could you know how many machines you have to be hooked up to? Because sepsis is multiple organ failure. Your heart goes, your liver goes, your kidneys go, your brain goes. And this is very expensive to treat. Well, versus just another media. I mean, it'd be insanity for not that to happen, and it will happen. Yeah. But this is different. These molecules have never been on the planet before. Okay, we don't know anything about them. What they are, they're these simple chemicals. They're like Legos, and they have many advantages over current antibiotics. If we had a time, I could show you antibiotics are really complicated structures. Bacteria are great chemists. We're not that great, actually. Vegans haven't been able to, catch up to them. Okay, and they're really hard to make, and they're really expensive to make, and they're really hard to alter in case you get a resistant bacterium. Now, and this is why these new fancy drugs like carbapenems and penicillins, these are changing penicillins to something else. It's really hard to do and really expensive. These molecules are like that. These COEs, they're just chemical compounds, modular chemical compounds like Legos. I got a red one, a blue one. Okay, does that work? No. Okay, well, let's put a yellow one in, and the synthesis is incredibly simple. It's high school chemistry, actually. Actually, we had a high school student make it just to prove it.
Wow.
He did it in a week too. So someone who had never been in a lab before did it, made a new COE and a milligram, a lot, which is a lot of antibiotics. Yeah. And so, but the issue for me anyway, the issue is going to be toxicity. Yeah, we've shown that, okay, in a week-long infection course, we show we can now affect the bacteria, we can cure it with the drug, and that was successful. We've also taken infected the animals, administered IV the drug to the animal without the bug, and they do fine. There's no visual, and typically, and as a control, we took drugs that are used in the clinic that are known to be highly toxic, and we could see that the mice didn't look fine. Okay, we showed that as well. But what happens after 30 days? What happens, across the population? What happens in a human? We have no clue because we haven't done the experiment. It's not a reason not to do the experiment, but I see that this is not an FDA-approved drug. This is something that's new. What does it do to the environment? No clue. However, the risk is worth it. I mean, here we have something that can cure every pathogen that we've tested. That's unheard of,
Yeah.
But the toxicity is going to be an issue, and we're working hard on that. It's going to be a long road. We're also going to have to need to know. That's number one, to really understand the toxicity. Number two is really understand the mechanism of action and resistance. We have a general idea. That's not good enough to get an FDA approval. You got to know what it does. You can't just get a thing. Oh yeah, we think it's the membrane. They'll never approve it. And so basically, at the end of the day, when we get that figured out, additional safety and efficacy studies are going to have to be done in humans.
Right.
That road is much longer than the test, but we think we can get there. We think that's. I tell you, I gave a talk at the NIH. A couple of them actually before the paper came out. We told them about this in advance, and I thought I was being pretty nice about sharing both the test and their drug before the papers came out. This was this year, and so I give this talk, and then the question session came up, and then the whole thing. Did you do this test, this in vitro, which means in the lab test, this test, this test, this test? And I was like, I go, what? Like, it wasn't. The message was go in the animal first. That was the message of them. That people aren't petri plates. That was the title of the talk. Well, okay. And then I fumbled. I didn't know what to say. And then the organizer said, I think what Mike's saying is that you should go in the animal first. Either the tests can be done at some other point down the road, but not. And then I just said, let's review what we've done. We didn't do any of those tests.
Yeah.
Okay, let's start with that. But what we did do is cure 2 untreatable diseases for which there are no molecules on the planet Earth. We did that,
Right.
and you can really see the disconnect that we need to look at things differently,
Yeah.
And however, to get funded for that, these are the guys that are on the panel.
Yeah.
It's just this fight. Well, I'll tell you, for the antibiotics, everybody has their stories, but for the antibiotic, the new test, I wrote that to the NIH like in 2009, I think. I mean, I got kind of laughed at. Like, like I said, here's the, what I got what's called triage, which means it doesn't even get discussed. Like, to be blunt, this grant sucks. That's what it means. Okay. And what I did, I provided the major targets, which was not that many. You target the cell wall, the DNA, the RNA, the protein, and I got a few essential vitamins. But that's the list. It's not long. And I made a list of this and said, hey, that we thought that we had a new test. It never made sense to me that the rich media would be the test. And what we got back was, this has been used since 1960. It's been very helpful, and you don't have to tell this esteemed panel the mechanism of action of these drugs. The point I was making wasn't that. The point was we don't have many. That list is really short.
Yeah.
You know what? I'm never going to get funded by them, and for this work. And so I said, we're going to get this done, and and I could say that also something about this show that's really helpful to get the word out is that there's also a disconnect in funding. Like, and the NIH has been very generous to us. The Army has been very generous to us, but they fund discoveries. And then the industry, they fund when it gets to a point where they can think they can, market something and make something.
Yeah.
Okay. There's a huge middle there. That's where we are with both the susceptibility test and the drug. The susceptibility test, we have to now do this, additive therapy and then the, replacement therapy and all that. Well, somebody has to fund that. Okay. It doesn't come out of, but it's not going to ever get to a market until that's done. And then with the new antibiotic, not until this toxicity stuff, and that's going to be a lot of money and a lot of time. Yet, you're in this thing. Oh, you've already made a discovery. You get it from the industry. And they're just going, no, we're not going to touch that till you get this stuff done.
Yeah.
So it's like this. It's like crazy town to me. Like
no man's land.
Yeah, it's no man's land. But the thing is, it's not no man's land. But we know it works.
Yeah,
it can work. It's that crazy town here. It's crazy that it's. We're struggling with that, but we're going to get there. It's nothing we can't handle. But I thought that. That road may be paved a little bit easier, but that's the hard part of this business, the academic business is that it's been whatever.
I have one more science question. So there must, or I won't say there must be. Are there hints in the ones that are toxic, the differences between those and the one that is not?
Yes, there are.
That tell you kind of what's going on.
You know what? That's a super good question, Chris. And what's really cool about these COEs is that it's really easy to do those experiments. You just change the Lego to, what I mean by you have
a building block.
Building block. You have an amino group versus a carboxyl group, whatever. It's just really easy to change it. And so those experiments are being done, and it is making some sense. It's not quite. It's nothing I can say, but we kind of know. Oh, well, we did. We published this, so this is important. So out of 17 antibiotics, 17 COEs, they're not formally antibiotics, they're antimicrobials, that only one of the 17 were not as toxic as the others. Only one of the 17 could kill both gram-positive and gram-negative, the same one. And then what we did, we actually did your experiment you just suggested. We changed the Legos a little bit and asked, can we make it better? And is it less toxic? And does it still retain the efficacy?
Right.
Those things have to be together, right? You can't just have one. They could work. Okay, it's not toxic, but then it doesn't work. Well, and the answer was we showed that by altering, they're the same class of COE, but we did pretty serious chemical changes, and we could show that when you're in, we found where the sweet spot was. Okay, and it just turned out that this 1 out of 17. It was within the sweet spot, and we would never ever have proceeded. But I really, this thing about this person dying of a UTI, it just kept bugging me. Like, we just have to try until the science says no. And we kept having these no moments. Okay, it just never stopped. so which is a good thing. So whatever. After 5.5 years, then we finally got there. But really, for the first 3.5 years or whatever, we didn't have a dime. We were just siphoning it off. It was taking half my time and half my guys' time. And so that means that we're not working on the stuff we're really funded for. But now everybody's happy that, they're all happy. The NIH is happy. The Army's thrilled, whatever. And it also shows that the importance of having, of the U.S. Army and the NIH funding basic research with a little flexibility, to make discoveries. I mean, we had monies to do other very important things. We work on sepsis. That's what basically most of the money we get is from the NIH to work on the molecular basis of sepsis, to understand the mechanism, and then also to make things, to make molecules that will stop this. And we're I'm part of this large sepsis group for over almost 10 years now to do that. So that's where a lot of the money came from, and a lot of money came from the U.S. Army. And they, I thought there was going to be a little bit of a disconnect, and that, well, this is not what we funded you for. And they were, very happy about it, and they were very, and so I thought, you know what, this has to get out there, that this should be rewarded. This behavior of, okay, yeah, if I knew, if this is my opinion only, so this is just for me. If I knew the answer to every question before when I was writing a grant, that's not that interesting to me.
Yeah.
Generally, if you already know, it's like those days are over. I mean, I'm toward the tail end of my career. It's like we want to make big steps, and so not the same stuff. And the problem is, it takes money to do it.
Yeah.
And 90% of my time, or whatever, 80% of my time is writing for money. It's like in the last actually 3 or 4 years, we've had the money and we had the guys, and it's been the most fun, because usually it's a cycle. There are cycles, they're opposite. Like, so you have an idea, but you don't have the money to do it. And then, then you finally get, you get the money to do it. And then you have to get the people. And so this wave that when they're together, I've been here 30 years. I think it's the second time in 30 years that the waves were together, and you make all these discoveries because you have the money, you have the guys, and the motivation, and you get it done,
Nice.
so that's really super fun. And now we're on fumes. Yeah, actually, I just started a sabbatical this month, actually, for a year, and all I'm going to do is focus on funding and getting this stuff. We're going to execute this stuff. We're not going until this is done. And
Right.
and so so for a year, we're just going to write for money. And so anyway,
Nice.
yeah.
Well, Mike Mahan, this has been a treat to reconnect and find out about all this exciting work. I mean, I think it's really cool what you're doing, and obviously new antibiotics and the change in the gold standard test, I hope. And hopefully we make some progress.
And I just want to say thank you, and have a forum to be able to let this stuff get out there. And it's the only way the change is going to be made. If it's just in some lab in some place in Santa Barbara, it's going to go nowhere. The glacier has to jump. It has to jump from one side of the iceberg to the other. And this is the only way it's going to happen. So we really appreciate your efforts in being able to get this. It's very important work you're doing, and not many people are doing it. So anyway, I wanted to thank you as well. So
Well, it's my pleasure.
Okay, great. Thank you.
You bet.